everyone, welcome back. Today's video will be about the 20 amino acids and how they are categorized. More specifically, this video will discuss the following: a brief intro to amino acids, what you need to know for the MCAT, methods used to classify amino acids, how amino acids are grouped into categories based on the nature of their R groups, acronyms and phrases to remember the amino acids for each category, ways to memorize the structure of each amino acid, and general tips to use if you want to make up your own acronyms or phrases to remember the amino acids for each group. A brief intro to amino acids. Amino acids are the building blocks that make proteins. Almost all amino acids follow the same basic structure. You can see that structure on the screen. They contain a central carbon atom. This is often called the alpha carbon, an amino group, a carboxylic group, a hydrogen atom, and a side chain. The side chain is often called the R group. There are many types of amino acids, some of which don't look like the structure that you see on the screen. However, the ones we need to know all follow the same structure that you see on the screen. Here you can see that the amino group and the carboxylic group are attached to the central alpha carbon. And because of this, these are called alpha-amino acids. Amino acids and the MCAT. All amino acids that exist do not necessarily follow the same basic structure like I mentioned before. For example, in some amino acids, the amino and carboxylic groups are not linked to the central alpha carbon. Some amino acids are not encoded by the genetic code, and others, they are not used to make proteins. However, for the purposes of the MCAT, we are only required to learn about the 20 proteogenic alpha amino acids. Proteogenic meaning that these amino acids are used to make proteins. If you are taking the MCAT, the structures and the abbreviations, that is both the 3 and 1-letter abbreviations of the 20 amino acids are important to know. They have been repeatedly tested on the exam. Therefore, it is important for you to memorize these if you plan to take the MCAT exam. Categorizing amino acids. The 20 proteogenic alpha amino acids can be categorized based on many traits. The following methods of categorization are the most common on the MCAT exam. Categorization method 1. This method is based on the properties of the R group. In this method, the 20 amino acids are grouped into one of 5 categories. They include the following: 1. Nonpolar aliphatic amino acids. This is also called nonpolar non-aromatic amino acids. 2. Aromatic amino acids. 3. Polar amino acids. 4. Acidic or negatively charged amino acids. And 5. Basic or positively charged amino acids. Categorization method 2. This categorization method is based on the affinity of amino acids to water. Within this method, the 20 amino acids are grouped into one of 2 categories. They include the hydrophobic amino acids and hydrophilic amino acids. If you want to learn more about the second categorization system, check out my video on this topic. I didn't want to include it here because that would have made a really long video. Let's get into the first categorization method and learn about the 20 amino acids and how they fall into each of the 5 categories. Group number 1, nonpolar aliphatic amino acids. 7 of the 20 amino acids fall under this category. They include the following: glycine, Alanine, Valine, Leucine, Isoleucine, Methionine, and Proline. As I mentioned before, you really need to know the 1-letter and the 3-letter abbreviations. An easy way to remember this group is by memorizing the acronym GAVLI-MP. This is what helped me to memorize these for my exam. However, if you want to make your own acronym, I encourage that fully. At the end of this video, I have highlighted some helpful tips that may be useful when you're generating these acronyms, or it could even be a phrase. Let's look at the structure for these 7 amino acids. First, we have glycine. This is the simplest amino acid that there is. The R group is a simple hydrogen atom. Because of this, glycine is the only amino acid that does not have a chiral center. This would be important for any type of biochem exam that you would have. Watch my video on chirality to learn more about chiral molecules. Then we have alanine. In alanine, the R group is a simple methyl group. This makes it easy to remember this. And as you will see, most of these amino acids kind of build up on each other. It's adding a group or changing the position of an atom. So it's kind of easy to remember these structures. Thirdly, we have valine. From valine onwards, the R groups start to become a little more complex. However, the way I remember valine is by seeing that the R group of valine looks like a V. 2 additional carbon atoms have been added to the R group of alanine to create valine by creating the shape of a V, and therefore that is valine. Then we have leucine. This is the L of GABLI-MP. The main reason why I like this acronym is because it helps me to remember the structure of the molecules it refers to. Like I mentioned before, each amino acid, as laid out in this acronym, builds on each other, and there's a minute change in each that creates the new amino acid. So, for example, in leucine, its R group is the same R group as valine, but extended by 1 carbon atom added near the alpha carbon atom. After leucine, we have isoleucine. The structure of this is easy to remember as well. For instance, consider redrawing the structure of leucine this way. If you do this, you can easily see that the main carbon chain of the R group has 3 carbon atoms, and then there's a methyl group. A methyl group is a CH3 group. This methyl group is attached to the middle carbon of the R group. If you remember this, then the structure of isoleucine is the exact same thing, but with the methyl group just moved up 1 carbon to the carbon that is closest to the alpha carbon. Then we have methionine. This is a little different from the previous molecules and is one of the 2 amino acids that has a sulfur atom in the R group. I don't necessarily have an easy way to remember it, but it's quite easy to memorize. And lastly, in this category, we have proline. How I remember this molecule is by noting that this is the only molecule where the last carbon atom of the R group interacts with the amino group of the amino acid, making it cyclic. The great thing about the MCAT is that it is a multiple choice exam, so you don't necessarily need to be able to recreate amino acids exactly as is. You just need to be able to identify the structure. Since this is the only amino acid that has an R group connected to the main amino group, it's quite easy to identify it. The same applies to methionine as well, since it's the only amino acid that has a sulfur atom in the middle of the R group chain, if that makes sense. You might understand what I'm saying when we talk about cysteine, which is the other amino acid that has a sulfur group. So this covers all the nonpolar aliphatic amino acids. Let's get into the second group, which is the aromatic amino acids. 3 of the 20 amino acids fall under this group. They include tryptophan, phenylalanine, and tyrosine. An easy way to remember this group is by memorizing the acronym WIFI, which is WFY, and pronounced as WIFI. I came up with this acronym and I used it because the acronym is made of the 1-letter abbreviations of the 3 amino acids. So let's look at the structures of these three amino acids. These are easy to remember and identify. This is because the amino acids in this group have a benzene ring. So once you see a benzene ring, all you have to do is to differentiate between the three. The first molecule is tryptophan. This is the most complex looking amino acid within this group. However, I learned to identify this by first making sure that the amino acid has a benzene ring. After that, if you see another cyclic ring above the benzene ring, then you know that this is tryptophan. Another thing that I have noticed about the MCAT is that they do not include made up or false fake amino acids as a response option for a question where you are required to identify a specific amino acid. This is at least true as of recording this video. So having a rough idea about how they each look should be sufficient for you to correctly identify these molecules. Then we have F of the Wi-Fi acronym, and this stands for phenylalanine. This is an easy molecule to remember. If you see an amino acid with just a benzene ring that is attached to the alpha carbon via a CH2 group, then this is phenylalanine. And finally, in this group, we have tyrosine. This molecule is also easy to remember because it's an extension of phenylalanine. The only difference is that it has a hydroxyl group, that is, an OH group attached to the benzene ring at the fourth carbon. This is also called the para position. Those are the only three amino acids in the aromatic amino acid group. Now let's look at the polar uncharged amino acids. 5 of the 20 amino acids fall under this category. They include serine, Threonine, Cysteine, Asparagine, and Glutamine. The acronym I use to remember this is STCNQ. Again, the acronym is made of the one-letter abbreviations used to identify each of the amino acids. This category can be a little challenging to remember, but it's completely doable with a little practice and effort. So let's get into it. I'm going to tell you the method I use to remember these amino acids. Firstly, I found commonalities in the R groups of these 5 amino acids. Then I figured out that there are 3 types of similarities between each group. So let's think of these as boxes, and we put in each amino acid into the appropriate box. So the first box would contain amino acids that have a hydroxyl group, that is an OH group. The second box will contain amino acids that have a sulfur atom in the R group. And the third box will contain amino acids that have an amide group. An amide group is a group where you have a carbon atom double bonded to an oxygen atom and single bonded to a NH2 group and another atom or group, as you can see here. Now that we have these 3 boxes, then we put the appropriate amino acids into them. So the 2 amino acids that fall into the first box are serine and threonine. This is because they are the only 2 amino acids in this category that contain a hydroxyl group. 1 amino acid falls into the second box. This is cysteine. This is because this amino acid contains a sulfur atom in the R group. And in the third box, we have 2 amino acids that go into it. These are asparagine and glutamine, and this is because the R groups contain an amide group. If you remember the acronym STCNQ, then the division of these amino acids follow the exact same method. You just need to know that the first box has 2 amino acids, second box has 1, third one has 2 amino acids as well. So now let's look at their structures more closely. Serine. The R group of serine has 1 carbon atom that is attached to the alpha carbon. This R group carbon atom has 2 hydrogen atoms and a hydroxyl group. Once you memorize this, it becomes easy for you to remember the structure of threonine. Let's look at threonine. As you can see, this too has an OH group, and therefore that's why we put it in the first box. And if you compare this to serine, you see that the structure is very similar. You get the structure of threonine when you add a methyl group to the end of the R group of serine. The great news is that only 3 amino acids of the 20 that we need to know contain OH groups as a part of their R groups. They are tyrosine, serine, and threonine. If there is no benzene ring, you automatically know that you can eliminate tyrosine, and therefore, from that point onwards, you basically have to differentiate whether it's serine or threonine. Now let's look at cysteine, which was the amino acid that we put into the second box. If you look at the structure of cysteine, you see that it has a sulfur-hydryl group at the end of the R side chain. Similar to amino acids with OH groups, amino acids with sulfur atoms are also relatively easy to identify because there are only 2 amino acids that have sulfur atoms of the 20 amino acids that we need to know. One is methionine and the other is cysteine. One of the quickest differences you see is that methionine has a longer side chain than cysteine does. And in methionine, the sulfur atom is found in the middle of the side chain, while in cysteine, the sulfur atom is found at the end of the side chain. Now that we have completed 3 of the 5 amino acids in this category, let's look at the remaining 2. If you remember, these 2 fell in the last box that we created, the box that contains amino acids that have amide groups. There are only 2 amino acids that contain amide groups out of all the 20 amino acids that we need to know. So it's kind of easy to identify them. Essentially, you only need to be able to differentiate between the 2 amino acids that contain the amide groups. So let's look at asparagine and glutamine, which fall under this category. This is asparagine, and this is glutamine. In asparagine, there is 1 carbon atom separating it from the alpha carbon, while in glutamine, there are 2 carbon atoms. This is the only difference. So if you are able to identify the structure of asparagine, then you are only required to add another carbon between the R group and the alpha carbon to know the structure of glutamine. Now let's look at the acidic or negatively charged amino acids. This group contains 2 of the 20 amino acids. They include aspartic acid or aspartate if it's deprotonated, and glutamic acid or glutamate if it's deprotonated. In normal physiological conditions, both would be deprotonated since they are acids. This is what gives each of these amino acids a net negative charge, making them negatively charged. The acronym I use to remember these 2 amino acids is D, as in DE, or ED. To be honest, I prefer ED. And it's because it reminds me of Ed Sheeran and how his album covers tend to have math symbols on them and therefore relating to the negatively charged amino acids. But you can use whatever acronym that you would prefer. Similar to asparagine and glutamine, the structures of aspartate and glutamate are easy to remember. In fact, the way they differ is exactly the same. The R group of aspartate contains a carbon atom attached to a carboxylate. As a refresher, a carboxylate is a carbon atom double bonded to an oxygen atom and single bonded to an oxygen that has already lost its hydrogen atom. So it's a C-double-O- group. A carboxylate is basically a deprotonated version of a carboxylic acid. The structure of glutamate is exactly the same, with 2 carbon atoms between the alpha carbon and the carboxylate as opposed to 1 carbon like in aspartate. This is the same difference you see between asparagine and glutamine. Now, this could be a little confusing because both kind of sound the same and the difference is the same as well. So if you think you might get confused, remember this. Asparagine and glutamine both end with "in." and therefore contains an N at the end of their name. These are the ones that contain the amide, which also contains a nitrogen atom, which is an N. I hope that little trick would help you to minimize the confusion. And finally, we have the last group, which is the basic positively charged amino acids. This group contains the last three remaining amino acids. They include lysine, arginine, and histidine. To remember these three, I had to come up with a phrase for myself because the acronym didn't work. The phrase I came up with was K-less run hard. I found that this helped me to equate the letter K as the one-letter abbreviation for lysine. If this doesn't work for you, feel free to make your own acronym or phrase using the tips that I've included at the end of this video. In normal physiological conditions, these three amino acids would be protonated since they are basic. This is what gives the amino acid a net positive charge, making them positively charged. All right, let's look at their structures. Lysine. Lysine contains one of the longest straight chain R groups. It contains 4 carbon atoms in the side chain backbone, and it ends with a nitrogen atom that acts as a hydrogen acceptor. Because it has this relatively long carbon chain, it might be a little easier to commit the structure to your memory because of the really long carbon chain and the nitrogen atom at the end. Next, we have arginine. The R group of arginine is similar to lysine in that it has a comparably long, straight side chain, but it also has 2 nitrogen atoms in the backbone of the side chain and 1 nitrogen atom double bonded to the last carbon atom of the side chain. Therefore, this is sufficiently different from lysine in order to differentiate the two and identify them correctly. And finally, we have histidine. Histidine is one of the easier amino acids to remember because it has a ring structure at the end of the R group. It does not contain a benzene ring, so you know it's not one of the aromatic amino acids. The other amino acid that may look somewhat similar to histidine is proline. Remember, the last amino acid of GAVLI-MP, the aliphatic amino acids. However, they are different in that the ring structure formed in proline is made by the R group forming a bond with the amino group of the amino acid, while the ring structure formed in histidine stands alone and is made of 3 carbon atoms and 2 nitrogen atoms. As I mentioned before, you don't have to memorize all these amino acids as you are not required to recreate them from memory. So what you need to be able to do is to see the structure and identify it. However, make sure that you know the structures in all the forms that chemical compounds could be represented. So you see the Lewis structure as line structures or as wedged dash line structures as well. And that covers all the 20 proteogenic amino acids that you need to know for the MCAT. Before I end the video, here are some tips for you to remember the amino acids better. Make an acronym or a phrase to remember the amino acids for each group. If you're making an acronym, consider using the single letter abbreviations to minimize confusion on exam day. If you are producing a phrase, make sure that the first letter of the word in these phrases refer to the single letter abbreviations to minimize confusion as well. Try to make acronyms or phrases that are personal to you, funny, and are wild. This helps you to remember them better. And lastly, use the acronyms and phrases often to make sure that you remember them. I used to play this amino acid matching game for a few minutes every day to make sure they were fresh in my mind. I'll include a link to that game in the description section below in case any of you want to try that out too. And that's it. I really hope this video was helpful for you. Thank you so much for watching it. Before you leave, please make sure to like and subscribe Both of those really help out the channel. Thanks again for watching. See you in the next video. Bye.